Hello everyone, welcome to another episode of our channel. In the previous video, we discussed about unicellular and multicellular organism, and there we mentioned two terms. The number one is prokaryotic cell and the second one is eukaryotic cell. Today we will discuss about the basic concept of prokaryotic cell. On the basis of organization of genetic material and other subcellular structures, Dougherty divided the cells into two groups. Number one is prokaryotic cell and number two is eukaryotic cell. Further, Dodge and other scientists mentioned another type of cell which is called mesokaryotic cell. Today we will discuss about the prokaryotic cell. The Greek term pro means primitive and karyon means nucleus. So we can say a prokaryotic cell is the one in which the genetic material is not organized into nucleus and all membrane bound organelles are absent, so that there is single envelope system of organization. Prokaryotic cells occur in bacteria, Cyanobacteria, Archaebacteria, and Mycoplasma. Characteristics of prokaryotic cell. In the prokaryotic cell, organized nucleus is absent. There is absence of nuclear membrane and nucleolus. DNA is naked due to its non-association with histone protein, presence of mucopeptide and peptidoglycan in the cell wall. Ribosomes are 70S in nature, which have two subunits, one is 50S and the other is 30S. Presence of mesosome, presence of plasmid in cytoplasm. Mitochondria, Golgi bodies, chloroplast and lysosomes are absent. Let's see the structure of prokaryotic cell. We can divide the structure of prokaryotic cell into two parts. Number one is cell coverings or cell envelope, which contains slime layer, cell wall and plasma membrane, and the second one is protoplasm, which contains nucleoid and cytoplasm. Slime layer. In the slime layer, mucilage or glycocalyx is present. It is non-cellulosic polysaccharides and polypeptides. Thick layer of mucilage is called capsule. A loose sheath of mucilage is called slime layer. The term microcapsule is used if it is thin. Mucilage protects these cells against desiccation, toxins, phagocytes and viruses. This is capsule and this is slime layer. It's all about the thickness. In the capsule, the thickness is greater than 0.2 micrometer. Cell wall. A cell wall is a structural layer surrounding the cell just outside the cell membrane. It can be tough, flexible and sometimes rigid. Cell wall is elastic, porous and freely permeable to solutes less than 10,000 Daltons. It provides the cell with both structural support and protection and also acts as a filtering mechanism. The main constituents of cell wall is mucopeptide and peptidoglycan. Peptidoglycan has two different parts in this cell wall. One is N-acetylglucosamine and one is N-acetylmuramic acid. Porine protein occur in this layer which allow the passage of low molecular weight hydrophilic substances. Plasma membrane or plasma lemma. It is thin, selectively permeable quasi-fluid membrane which forms the innermost component of cell envelope and outer covering of cytoplasm. Plasma membrane has typical lipoprotein structure as visualized by fluid mosaic model. There is a lipid bilayer with extrinsic, intrinsic and transmembrane proteins. Periplasmic space occurs between plasma membrane and cell wall. Mesosome is an extension of plasma lemma. Mesosome is a convoluted multilaminated membranous structure which is formed by invagination of plasma membrane. Nucleoid. It is folded single circular strand of DNA double helix which is also called genophore. DNA strand is 250 to 700 times the length of the cell. It is folded in a supercoiled manner with the help of non-histone proteins or polyamines and RNA. Prokaryotic DNA is called naked due to its non-association with histone proteins and absence of nuclear envelope. Plasmids are additional or extra-chromosomal small rings of DNA, having a few useful but non-vital genes. Plasmids have these days become an important tool in genetic engineering as vectors of genes. Cytoplasm. It is granular crystallocolloidal complex that fills the cell protoplast but excludes nucleoid. Sap vacuoles, cytoplasmic streaming and membrane-bound organelles are absent. There is presence of mesosomes, ribosomes, chromatophores in cytoplasm. Various types of inclusion bodies are found in cytoplasm. For example, gas vacuoles, which is found in cyanobacteria, inorganic inclusions found in many bacteria as volutin granules, sulfur granules, food reserve granules present in cyanobacteria as alpha granules, beta granules, etc. This is the picture of prokaryotic cell and this is the cytoplasm. Here this is the granules and this is the ribosomes. Flagella. It is microscopic hair-like organelle used by cells and microorganisms for movement. The word flagellum in Latin means whip. Just like the whipping motion flagella often used for locomotion. Bacteria flagellum has three parts: basal body, hook and filament. Basal body is rod-like basal part of flagellum which is occur in cell envelope and is attached to it by means of two pairs of ring in bacteria. Hook is curved and thickest part. It is made up of protein units different from those of filament. Filament is long tubular structure of flagellum. This is the basal body. This part is called hook and the rest other is called filament. It is made up of flagellin protein. Pili. These are filamentous projection on a bacterial cell used not for motility but for adhering to other bacterial cell or to animal cells. Pili are made of a distinct protein called pilin. They take part in conjugation. We saw the structure of bacterial cell. The structure of blue-green algae also possesses cell wall, cell membrane, capsule, ribosome, mucilage layer like bacterial cell. But apart from these, they have thylakoid, centroplasm or central body, pseudovacuole, chlorophyll, xanthophyll, and various type of other pigments. In this video, we discussed about the prokaryotic cell, the characteristics of prokaryotic cell, the structure of prokaryotic cell. In the next video, we will discuss about eukaryotic cell. If you have any question regarding this topic, mention it into the comment box or mail us. If you like the video, press the like button, share the video with your friends and subscribe our channel. Thank you.